There's no Reese's.
No Smarties either? That's all right. I think we're through
We're
that
all
one.
out here.
Oh. That's right. You all have a classmate who earned you a bonus point this afternoon. 1 bonus point for bringing me an outrageous bar. You don't all get to bring me an outrageous bar for a bonus point. The outrageous bar bonus point has been credited to Courtney. Thank you. It was delicious. Did we do tissue types? And so that here's an example of a common type question from me, a multiple choice question. I might say, which of the following is not one of the tissue types in the human body? I would list those 4 with one that sounds really good but isn't one. Okay, now I think I mentioned to this class that one of the most important steps, if you want to succeed on my exams, one of the most important steps is to be able to memorize stuff. And the stuff you've got to memorize is in the handouts. Okay, I want to visit with you after class about the book. It looks like you've got it, so maybe not much to visit about, but I want to tell you what I told them the first night. Okay, test yourself on memory. Know 2 things: how long it takes you to memorize stuff, and your brain is pretty good at telling you when you have something memorized, but you have to learn that. So it's like a lot of you might be, and I don't know a lot of you at all, and some of you I have a pretty good idea, but some students will sit with this and be talking to someone and looking and checking the score of the game over here and looking, talking some more score checks, looking. And after, say, 3 hours of that says, well, I've been looking at this for a while, I think I've got it memorized. Is that person got it memorized? No. And now I'm kind of, maybe exaggerating a little bit, but when you memorize something, even if it's a little list like 4 tissue types, which believe me is kitten's play compared to what you're going to want to absorb into your brain during the course of this semester, do it for real. Memorize them, then go with a blank sheet of paper into another room where this doesn't exist and write them. And make sure you can do that. And some of you can do that very quickly, very easily. Others of you, you're like, oh my God, I just looked at them, I said I had to memorize it. It's like nervous and Okay, if you don't memorize stuff when you come in to take my exams, it will be a complete and utter disaster, grade-wise. It doesn't mean after, say, exam 1, you can't fix it, but I would hate for your first lesson in how to memorize to be in preparation for exam 2. I want it to be in preparation for exam 1. Okay? And for those of you who don't believe I'm serious, exam 1 is about a week away. We'll see how it goes. I'm rooting for you all, but I've given out that message. You want to know the stuff that's in these handouts. Okay? We are then on to metric prefixes. Did we do that?
Yes.
Okay, and does anybody have questions on metric prefixes? Did everybody spend a little bit of their weekend on metric prefixes? Nobody, or? Wow. All right, I guess you all know. Alrighty then, let's see. I've already been requested to review the sodium-potassium comp, which I'm going to do. But so everybody in here, then, since nobody's asking me a question about metric prefixes, if I say 7.3 centimeters equals how many megameters, everybody could do that. Okay, because that's the kind of problem. When I list those prefixes, and I had you all add in centi and deci, are those the only 2 I had you add in? Yeah. Did I have you cross any out?
Yeah.
Nano and giga? Yeah. Good, yeah. How much memory do your, anybody got a USB port? A disk, portable disk, anything? Phones? You all got your phones? How much memory is on there? 128 gigabytes? Yeah, big phone.
256, so like.
Whoa, look at you all. Man, I'm still in the world of 16.
That's like 5 pictures nowadays.
Hey,
You have kids, you have to take a lot of pictures.
Yeah.
do you all know what a floppy disk is?
Yes.
Do you know what a 5.5-inch
Yes.
floppy disk is? I still have one, and it literally is floppy. And I remember when they came out with the 3.5-inch discs, we were all like, "Oh my God, you could put 5 Microsoft Word documents on this. That's huge." Kids, it's fun. You all talk gigs, you talk gigabytes. They're talking terabytes now. That's all this is. What's a gigabyte?
1/1000th of a terabyte.
It is. That's exactly correct. 1/1000th of a terabyte. What else is a gigabyte? How many bytes is it? It's a billion. A megabyte is a million bytes. That's what our floppy disks had, I think. It might have been kilos. Kilo is 1,000. Mega is 1,000,000. Gig is 1,000,000,000. Tera is 1,000,000,000,000. You all need to be able to navigate that number line because it's the easiest numbers to deal with. Powers of what? 10. And we like 10. 10 is our friend. It's all the other numbers that make stuff hard. Right? Okay. So, how do I do this? What's the answer? Anybody? So there should have been questions on the metric prefixes, maybe. Okay. How many zeros do I add? Lily, are you adding? Are you telling me how many zeros? And did you hold up? You say 8.
Oh, no, it's 8 less. So you add 7 zeros. Other way.
That way?
Okay, left.
Okay. Because centimeters, here's 7.3 centimeters. About, I don't know if that's exact, but that's about, okay. Can you even fit 1 megameter in there? No. So, good. 8 decimal spaces is correct, but you all also need to know which way do I move it. And the way to move it in this problem is this way. 8, and fill in with zeros, and that's how many megameters 7.3 centimeters is. That little line, did I draw the line on the board the other night? Okay, centimeters would be on the right side of your unit, megameters would be over here, and that's your clue that you need to go left in order to get to mega or to mega from centi. Okay? Okay, now I know some of you are good with this, because we did it in A&P, we did it in micro a little bit, and then we did it again in A&P and micro. So I'm putting it on the exam. If you don't get it, you have got to tell me and come up after class or come in early and we'll come up here 10 minutes before class and work on it. Okay? But if nobody asks questions, I figure, cool, you'll nail these 2 questions. Sodium potassium pump. I believe I drew a picture of that the other night and went on and on and on about how important it is and how much oxygen you expend for this pump. Right? Like how much? 2/3, yeah. That's no lie. Google it. Or well, actually not our textbook, but we have this really good A&P textbook. I think they mentioned that. Right? And then did I draw something like this? It kind of looks like a swing door with 3 sodiums going out and 2 potassiums coming in. This is my cell membrane. I'm getting quicker and quicker with the drawings. I know you all are oriented. Okay? And so eventually I've just got a bunch of sodium out here. Okay? Where does the sodium want to go? Yeah, it's crowded out here. It wants to go back in. Well, maybe we'll let it. And I got a bunch of potassium in here. Where does it want to go? Yeah, maybe we'll let it. Okay? Is that an equal exchange of charge? No. Where did more positive charge go? Out. So I can definitively say that across this membrane, it is more positive outside than inside. Okay? That's all that means. And what's the word for that? Begins with P. Good. That's polarized. That's all polarized means. That across a real or imaginary border, you can definitively say there's a charge difference. Okay? And this pump, this little protein called the sodium-potassium pump, its formal name is the sodium-potassium ATPase, but I just like calling it the sodium-potassium pump, and that's what I'll call it in your questions on the exam. It just sits there and does this all the time. And the reason it does this all the time in every cell in your body is because your cells cannot do their action, whatever that might be, without this setup. Because no matter what kind of cell it is, all I have to do to get that cell to do its action. Here would be an example of a cell's action. What do muscle cells do for me? They contract. Yes, I've got to do something to get it to contract. What does a nerve cell do for me? Secretes. What does it secrete? Neurotransmitters. Yeah, it shoots out neurotransmitters. That's transmission of a signal, but it really is just secreting. I've got to do something to get it to do that. What does a melanocyte do? Makes and secretes melanin, our skin pigment. These are just example actions of cells. What does a neutrophil do? It's a white cell. What does it do? By doing what? It eats stuff. You're not supposed to be here, get in my belly. That's what a neutrophil does. And to a bacteria, that's really scary. To you, that may not seem scary. But to a bacteria, a neutrophil can eat like 50 bacteria in one bite. It's like me with Reese's Pieces. Or M&M's. The other tasty chocolate candy. I'm totally off topic now. What am I thinking of? I just got hungry. Okay, so this cell is ready to do an action. And in order to get this cell to do its action, all I have to do is open a bunch of sodium doors. Sodium only. What's happening? Sodium will naturally go in, down its concentration gradient. That's called simple diffusion. It happens faster than you could ever imagine. And what charge is sodium bringing in with it?
Positive.
Is that going to turn that around? Am I polarized anymore?
No.
No, I am in fact, what word did we use?
Depolarized.
Depolarized. And in fact, depolarize is exactly what you need to do to every cell in your body to get it to do its action. You don't consciously do it, obviously. Okay? But that's how cells work. This charge difference also has another name. Oh, wait, let's finish this story. Let's finish the story. Immediately after that, I want to go back to being polarized. What's the quickest way to do that?
Some potassium.
Yeah, let's open some potassium doors. And where does potassium want to go?
Out.
Out. Bringing positive charge with it. Will that put me back to this?
Yes.
Okay, so I'm repolarized. And who's a little bit grouchy?
ATP.
Yeah, I just backwards to everything it was doing. But you know what? The sodium potassium pump, pretty good sport. As long as you provide it with ATP, a ton of it. 3 sodiums out, 2 potassiums in. 3 sodiums out, 2 potassiums in, it will get caught up. I've got a magic marker cap in my hand that weighs probably less even than a gram. Think this is hard?
No.
It's not. It's not. It's feather light, literally. Okay. Could I do it forever? Didn't we have this conversation the other night? Okay. Now, every single time I do this, I have to contract or shorten all the muscles that flex my elbow, that participate in the action of flexing my elbow. And every time I do it, they need to be depolarized and repolarized, and depolarized and repolarized, and depolarized and repolarized for every time. So there are limits to this, because even with this marker cap, we all decided that not even The Rock could do this for an entire day. Okay? The other night I was going to use Arnold Schwarzenegger, but I knew I would be dating myself. Then I'd have to talk like the Terminator, and I'm not really good at that. So we stuck with The Rock. Okay? Sodium potassium pump. I believe you asked about that, Vanessa. Do you feel a little better about it? Good, good. Those are the questions I'm allowed to ask you about. I think I did leave out one thing, and that's the term resting membrane potential, which I believe I called this when the cell is at rest. Good? Did we do that term the other night? Resting membrane potential. So here's how I might ask that. What protein is responsible for the resting membrane potential? What would be the answer to that? The sodium-potassium pump, yeah. Or I might say the resting membrane potential describes a blank charge outside the cell and a blank charge inside a cell at rest. What's my first blank for outside? Positive, and second blank is negative. I think I did that one with you all the other night. Okay? Sodium-potassium pump. All right, good. Let's do some genetics then. Since our brains are loose. That was a little stretch out session. And now for the workout. How it goes. DNA, one of my favorite things to talk about. Deoxyribonucleic acid. Let's not go back through all the steps and the game of replication, transcription, translation. Is replication related to transcription and translation? I mean, in other words, does replication have anything to do with transcription and translation? No, nothing. What do cells spend most of their time doing? Replicating or transcribing and translating? And it's not even close, by the way. Transcribing and translating. Let me compare. Why would I replicate my DNA? If I'm a cell and my DNA is in the nucleus, I'm a human cell, so I've got 46 chromosomes. That's my DNA in a nucleus of the cell. Why would I copy that? What must I be about to do? Divide, mitosis or meiosis. That's the only reason to replicate DNA. There's no other reason to copy your DNA. So let's kind of compare this to humans. I asked this in an A&P class. I said, who of you think you have more brothers and sisters than anybody else? Anyone think they probably come from a family that's bigger than most of the rest of us? Yeah, how many?
We have 4 sisters and 3 brothers.
4 and 3, so a total of 8. That's pretty good. That beats me. You?
Well, they're not all biologically, but I have 5 brothers and 4 sisters.
9, 8, 9. Okay, let's, and for the purposes of discussion, let's pretend it's all the same mother. Okay, but those are big numbers. I had one in A&P last semester that came from a family of 11. I thought, wow, that poor mother. So, okay, so a woman, let's just say a woman has 9 kids. Okay? A woman has 9 kids. Did she spend most of her life pregnant or not pregnant? Most. Okay, so 9 kids times 9 months, that's 81 months, which is about 6 years? 6.5 years? She lives till 75. So I ask you again, does she spend most of her life pregnant or not pregnant? And it seems like it, because we're like, oh my God, 9 kids. What else are you doing, right? A lot. And most of your cells that divide, unless it's in really, really, really rapidly dividing tissue, which oftentimes means there's a problem, then your cells spend most of their time doing transcription and translation. So what is the point of transcription and translation? What's the product? Begins with a P, ends with proteins. Proteins, yes. Because proteins are the tools cells use to do their jobs. And most people spend most of their lives doing their jobs, right? Same with cells. Replication is separate. You only do it when you're going to divide. It's a full copy of DNA. All 46 chromosomes in a human cell copied. So how many do we got now? I copy all of my 46 chromosomes in a cell about to divide. How many chromosomes are in there before I divide? 92. I know I asked you to do math in your head really quickly and you had to carry the 1. I know, really hard. Me too. I've just done this lecture enough to know it's 92. And then when I divide those, how many does each cell get? 46. Good. Okay? DNA is large, double-stranded, always in the nucleus, and has the nucleotides A, T, C, and G. These are just quick little bullet notes from a game you all played either with me in A&P or in another A&P class or in a biology class, where we unwound the DNA and we put a bunch of A's and T's and C's and G's and we matched up the complements. We replicated. And we learned the enzymes involved and all that business. We're not going to go back through that. We're past that. You've demonstrated that you all can move forward having learned that. Okay? But I definitely want you to remember these distinctions between DNA and RNA. RNA is smaller. RNA is single-stranded in humans. Okay, there are some rare exceptions to RNA being single-stranded in the microbe world. RNA can be either in the nucleus or the cytoplasm. It spends its life going back and forth sometimes, and it uses 3 of the same nucleotides, but we change the T to a U for RNA. If I want to ask you questions about DNA or RNA, number 1, they will not be trick questions, and number 2, they will be able to be answered from those 2 lines. Okay? Replication. You should definitely know replication is the process of using one double-stranded DNA to make a full copy of itself. So you go from one double-stranded DNA, that was the template, the one you used to make the copy, to 2 double-stranded DNAs. Since we're playing around with DNA, where must we be? Nucleus. Good. Now, if you want, put a little arrow from transcription to translation. Because while I said these have nothing to do with replication, they have everything to do with each other. Okay? Transcription first, then translation. And here's why. Here's the simple reason why this is 2 steps. What's the recipe book for proteins? DNA. Where is it? It's in the nucleus. The ribosomes are out in the cytoplasm. And who makes proteins? Ribosomes. Some's missing. How about if you asked your mother for her awesome chicken casserole recipe, because hers, of course, is always the best, right? And you say, hey, could I have that recipe? And your mother says, I'll give you that recipe, but when you cook it, I'll write it down on this piece of paper, and when you cook it, I want you to leave this piece of paper in your bedroom, but I want you to cook the casserole in the kitchen. Does that make any sense? Does that sound a little bit foolish? Is it sounding even a little bit weird? But since DNA cannot leave the nucleus, that's the recipe book. So all transcription is copying the recipe for one protein out of that big gigundous book in the nucleus, and taking that copy, what's that copy called? Begins with a little m. mRNA, or messenger RNA. That's allowed to go out into the cytoplasm, and it has a coded recipe right from the DNA. The ribosome grabs it, and that's the beginning of translation, which another term for translation is protein synthesis. One amino acid at a time. The ribosome reads, you all remember this, reads a codon, the amino acid comes. Then it goes to the next codon, the amino acid comes. Okay? So, those little notes that I put in your handout, that's as much as I'm allowed to ask you about replication, transcription, and translation. Okay? Will not ask you to do any complementary base pairing in my exams in Patho. So how many chromosomes in human cells? 46. What is the exception that is allowed to have 23 chromosomes? Gametes. In fact, they have to have exactly 23, right? Because remember, boy gamete fertilizes girl gamete. That is the first cell of the new person. How many chromosomes does that cell need to have? 46. So you got to have 23 in each. We're going to talk about some conditions where that is off a little bit. Okay. What's another? Think a little outside the box here. What would be another normal situation where you have a different number of chromosomes in a human cell than 46 or 23? Is there any other situation where you look at a human cell that has a different number of chromosomes and you say, well, that's normal? Okay, sit? No, that's not normal. That's not normal. That's a pathologic condition. Good, you're right, it's a different number, but that's not a normal condition. So where you would say, oh, there's blank number of chromosomes in the cell, blank is not 46 or 23, but you're not worried about it. We actually kind of just talked about this. Okay, do I make blood cells every day? How do I make a new red cell? Okay, in the bone marrow, that's where, that's not how. In one word, how does one red cell become two red cells? Mitosis. Mitosis. And what do you have to do in mitosis? Replicate. And a normal cell that's beginning mitosis replicates its DNA. How many chromosomes in that cell? We just did the math. 92. Is that abnormal? No, as long as it divides into two 46 chromosome cells. Yeah, that's a normal number in some of your cells. Now, would I be worried about that in an adult muscle biopsy? Yes. Because how many new muscle cells do adults make? Zip. You're done with muscle. You can make your muscle cells bigger, but you can't make more of them. Okay? You can keep some of your nerve cells from dying, but you can't make more of them. Reminds me of an old "Don't Do Drugs" commercial they used to run. Are any of you old enough to remember the fried egg?
Yeah.
Or didn't they run that for a long time? This is your brain, and this is your brain. First of all, I hope my brain is better than an egg. Okay? Because that's not it. This is your brain. Wait a second. I'm offended by that. Okay? Did you all know that you can, some people can kill a small number of brain cells occasionally when they hold in their sneeze? Which is kind of the polite thing to do. You know, you're in an elevator that's packed with people and you really got to sneeze. Do you go, huh? Do you do that to everybody in the elevator? Or do we go, huh? Do we do that? And we're like, oh my God, I hope nothing came out. Right? Let it out, people. Let it out. Just bury your face in your arm and let it out. Isn't that what we're supposed to do now when we cough? Cough on your arm. Say again? Yes. Yeah. Coughing and sneezing now. I know. Now you're just like, oh, I don't want that on my shirt. Hang on. Everybody in the elevator. I never did that. But I now let my sneezes out. I can't. Anyway. Rolling right along. Let's learn some terminology of genetics before we get into some. The first diseases we're going to talk about are actually genetic diseases. And by far and away, it isn't even close. It's like 99.9% of genetic diseases are not about the wrong number of chromosomes. They're about one gene is mistaken. So one protein recipe. The whole rest of that chromosome is fine. But one protein made by that chromosome is made wrong. That is what about 99.9% of genetic diseases are. Like, for instance, cystic fibrosis. One protein made wrong because one part of the DNA is wrong, on chromosome 7. All the other chromosomes and the whole rest of chromosome 7 is fine. Okay? But we're going to actually start first with some terminology, and then we'll talk about some of the diseases we see when the numbers of chromosomes are off. So let's start with genotype. Genotype. Genotype is the composition of genes at a particular site in a chromosome. In other words, you look at, say, chromosome 1. This isn't, I'm not going to be really accurate. This is kind of pretend play, because I don't know all of the functions of our chromosomes as well enough to do this perfectly. But let's just say chromosome 1 has eye color, hair color, and height on it. Okay? And certain eye colors have certain nucleotides, and certain hair colors have other nucleotides. Basically what genotype is me looking at somebody's chromosome number 1 and saying, here are the nucleotides for the I genes, here are the nucleotides for the hair genes. I'm literally listing the A's and the T's and the C's and the G. Genotype. I'm literally saying, what are the letters on those DNA segments at those chromosomes? Genotype. But phenotype is what those things make you look like. Like if on chromosome 1 your parents gave you blue eyes, blonde hair, and you're tall. That's your phenotype. You have blonde hair, you have blue eyes, and you're tall. I just described everybody on the Swedish swimming team. Seriously. Again, Google it. Okay? Phenotype is what the protein recipes on your chromosomes make you look like. And you know what? It's not just blue eyes, blonde hair, am I tall? And by the way, eye color, hair color, and tallness are way more complicated than just appearing on chromosome 1. But it's also, what's my blood type, okay? And things like, what's my skin color? How much melanin do I make? Everything that contributes to your physical existence is your phenotype. A karyotype is literally just laying out somebody's chromosomes and looking at them through a fancy microscope, so that you can see 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, 2 chromosome 4s. You want to see pairs of 22 autosomes, those are your non-sex chromosomes, and then of course either 2 X's for a female or an X and a Y for a male as their sex chromosomes. Autosomes, again, are your non-sex chromosomes. Their numbers 1 through 22. You have 2 of each, one from mom, one from dad. That accounts for 44 of your 46 chromosomes in a typical human cell. Your sex chromosomes, again, either 2 X's, X from mom, X from dad, or an XY, X from mom, Y from dad, are the last pair. So a lot of you, when I ask you how many chromosomes does the human cell have, a lot of people want to automatically say 23. The answer is 23 pairs. The actual number is 46. Pedigree is a chart that tracks trait expression. I will not ask you all to do this, but some of you may have done it or seen it done. It's when they do like a family tree, and they say, here's grandpa and there's grandma, and they had 1 boy and 1, 2, 3 girls, and grandpa was a carrier of this disease, okay, and it turned out that one of the girls and the boy were also carriers of the disease. And they all went off and got married, and they had kids. And this daughter happened to marry another carrier of the disease, and it turns out their first daughter has the disease. Okay? So this is kind of how we track, gee, grandmother and grandfather didn't have the disease, none of their kids had the disease, but one of the grandkids had the disease. A pedigree is a little family tree. If we know about the people's medical history, where we can kind of track the spread or the distribution, really. It's not really the spread, that's more like an infection. The distribution of genetic diseases within a family tree. Okay? Some of this terminology will make more sense to you when we actually talk about autosomal dominant and autosomal recessive diseases. Locus. Locus and allele. These are very difficult terms. So why don't I erase this and we'll try to do this carefully. Locus. Let's again pretend that chromosome 1 is entirely responsible for eye color. So I got a chromosome 1 from my dad, and I got a chromosome 1 from my mom. Okay, and let's just say that this part of those chromosomes is for eye color. Okay? Then we call that that part of chromosome 1, we call that the locus for eye color. And that won't just be in me, that will be in everybody. Okay? We now know that that is the location or locus for all of the eye color genes. Do we all have different colored eyes? Okay, so what an allele is, alleles are the different things you can have at that locus. Again, I'm simplifying it a lot, but let's say Dad passed on brown eye genes to me and Mom passed on blue eye genes to me. Okay? They're still, those eye color genes are still in the same locus or location, but there's different information there. Dad gave me the genetic information for brown eyes, Mom gave me the genetic information for blue eyes. So the alleles, the actual information at that general locus, is different in my parents. Let's say the same is true for hair color. Hair color is determined here on chromosome 1. And let's say Dad gave me the genetic information for brown hair, and Mom gave me the genetic information for brown hair. What color of hair am I likely to have?
Brown.
But can you predict what color my eyes will be?
No.
No, it may be a 50-50 shot. Maybe it'll be a combination of those. Okay? But so, let's get the first two terms down first. Locus is what location on what chromosome is certain information found. Like eye color and hair color, whether it's real or not. Alleles is what actual genetic information is at that locus. Okay? Then we move on, I'm going to jump down a little bit to homologous, homozygous, and heterozygous. Okay, the three H's, as I call them. Here's somebody's chromosomes, 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, ... and then eventually 2 chromosome 22s, and let's say this person is a boy, so they've got an XY. Okay, you all can fill in the dots, it's pretty obvious. Okay? Homologous chromosomes are the ones of the same number or the sex chromosomes. So chromosome 1 from Dad and chromosome 1 from Mom in all of our cells, we call them homologous chromosomes. They're like, partners. They hang out. Okay? Chromosome 2 from Mom, chromosome 2 from Dad are a homologous pair in our cells. Etc., etc., etc. And then whether you've got 2 X's or an X and a Y, your sex chromosomes are also called a homologous pair. That's what the term homologous means. Do you all know that you all really aren't a mix of your parents' genes? You're a mix of your grandparents' genes. Do you all know that? Did you know that before? I know, everybody's like, oh my God, you just spun my world backwards. You just tipped over my North Pole. Okay, it's not that big a deal because before you thought your parents were composed of your grandparents' genes, so you're getting them indirectly anyway. Your parents' chromosomes are in your gametes, and your gametes are your reproductive cells. But they arrived in your gametes the same way they look in you. A chromosome 1 from Mom, chromosome 1 from Dad, chromosome 2 from Mom, chromosome 2 from Dad. But in gametes, something really, really weird is allowed to happen. What's allowed to happen is homologous chromosomes are allowed to get all touchy-feely with each other. And when chromosomes get touchy-feely with each other, they're all sticky and friable. That means they break easy. So when chromosome 1s start getting all touchy-feely with each other, pieces of this chromosome get stuck to this one, and pieces of this chromosome get stuck to this one, and then your gametes are made out of that. So now your gametes have 2 chromosome 1s that never existed before. They're a combination of the chromosome 1s that your parents gave you. But those are going to your kids, not to you. They're in your gametes. So really the diversity step and the family step is 2 generations apart. Again, that'll become a little bit more clear as we flow through this. Now, homologous, everybody clear with that? Homozygous and heterozygous refer to loci. So here I am back at chromosome 1. I have an eye color locus that has different alleles, brown and blue, and a hair color locus that has the same alleles, brown and brown. The two that are different, when two alleles at homologous loci are different, we call them heterozygous traits, and here we call them homozygous, the same. Okay? So when your parents both gave you the same information, those are homozygous alleles at those loci. Meaning here, you all pretty much kind of got the message here when I said, if this is what I got from my parents, what color is my hair likely to be? Probably brown. So when you have homozygous alleles, you're less likely to have much different than from what's there. But if I got heterozygous alleles, it's kind of a flip a coin. Blue eyes, brown eyes. Okay? Tough terminology. Tough terminology. Spend a little time with it. Let's go back up to euploid. Euploid. U. Sorry, I'm just having fun with it. That prefix usually means normal. Normal. EU. Like, you may have heard the term euthyroid. That means somebody's thyroid gland is running normally. It's not hyper or hypo. EU is a prefix that means normal. Ploid means number of chromosomes. So euploid means normal number of chromosomes. Typically in human cells, we have 3 types of normal. Our gametes are haploid. They have one set of 23 chromosomes, hap1. Our rest of our cells, like 99.8% of the cells in our body, are diploid. 2 pairs of each chromosome for a total of 46. And finally, we have those rare situations where you're looking at a cell that's in the just completed replication. Again, you would see a lot of these in bone marrow. You would see a lot of these in somebody's stomach epithelium because it replaces itself every 3 to 5 days. But other than that, we're really not actively growing in our tissues as adults. Okay? But tetraploid, 4 sets of 23 chromosomes. That's a total of 92. All of those can be considered a normal number of chromosomes in the right context in the human body. Polyploid means a cell has an abnormal number of chromosomes by a factor of 23. Okay, so here would be an example of that. How many chromosomes is a gamete supposed to have? 23. So a gamete with 46 chromosomes is a polyploid cell. Not only does it have too many chromosomes, it has exactly 23 too many. Or a muscle cell. How many chromosomes are my muscle cells supposed to have? 46. A muscle cell with 69 chromosomes is a polyploid cell. It's off by exactly 23. Aneuploid is off by not a factor of 23. And the factor we're usually talking about here is 1. We're usually talking about off by 1. Like a cell with 45 chromosomes, or a cell with 47 chromosomes. Both off by 1. Okay? Aneuploid, off by not a factor of 23. Dominant traits. Dominant traits. Remember, every trait that for the most part, until we learn otherwise, and we will learn a couple of exceptions to this, but basically, since I'm getting a chromosome 1 through 22 and a chromosome from my mom and a chromosome 1 through 22 from my dad, that all the information on chromosome 1 through 22, I could go with my mom's genes or with my dad's genes. Everybody kind of cool with that? And we're kind of a combination of that expression. Okay? Well, if mom gives me, let's say I'm on chromosome 1 again, and I get a tall gene from dad and a not tall gene from mom. There's really no such thing as tall genes and not tall genes. Again, I'm way oversimplifying it. Okay? But let's say this is how it went down for me. I got a chromosome from dad that says genetic information for tall, and chromosomes from mom that do not have genetic information for tall. Okay? I call that not tall, and that's why I use a small t rather than a capital T. If tallness is a dominant trait, then all you need is one of your parents to give it to you, and you will be tall. So if tallness is a dominant trait, and this is a realistic scenario for what I got, I will be tall. On the other hand, if tallness is a recessive trait, you need both parents to give you that gene in order to express it. So will this person be tall if it's recessive? No, they didn't get both. They got tall here. Can they pass a tall gene on to their kids? Yeah, does anybody know what that's called? This person then therefore is a tall carrier, a tall carrier. You only have a carrier state when we're talking about a recessive trait, okay? And all that means is I don't have that trait or disease, but I could pass it on to my kids, okay? Dominant versus recessive there. A few more terms, then we'll talk about some conditions. Translocation. Translocation. Remember how I said that at a certain time of development and gamete progression, that chromosome 1 is allowed to touch chromosome 1 and exchange material, and chromosome 2 is allowed to do it with the other chromosome 2? But that's it. You can only touch your homologous pair. A translocation is when that rule is violated. Like when chromosome 4 exchanges material with chromosome 17. That's not allowed. Chromosome 4 is only allowed to get hands-on with the other chromosome 4. Okay? A translocation is a pathologic condition in which two non-homologous chromosomes have shared information or have exchanged information. A Punnett square. Punnett square. Some of you may have done this. We'll only do a quick 2x2, and the math should be really, really straightforward. Here's how a Punnett square works. You put, say, mom up here, and you put dad up here. Whoops. Dad over here. Okay? And we say, let's just pretend tallness is a very simple hereditary trait passed on, okay? And we look at, and let's say right now it's a dominant trait. And we look at a male and a female that are going to have kids. And before they even get pregnant, they want to know, oh my God, are our kids going to be tall? Because we're big basketball fans. Okay, and you tell them, well, tallness is a dominant trait, but until we look at your chromosomes, we don't really know if your kids are going to be tall. So the parents say, well, why don't you take a look at our chromosomes? And they say, okay. And they look at dad, and dad has that from his dad and that from his mom. And then the prospective mother has those from her parents. Okay? So, is dad, from the information I've just given you, is this guy tall?
Yes.
Yes. Why? What's the key piece of information there? I said it was dominant trait. Is the prospective mom tall? No. Okay? Now, what a Punnett square is now that we know this, now that we know all of the potential alleles that these parents can pass on to their kids, these boxes are possibilities for their kids. Okay, so for the first box, I take what is next to it from mom and dad. So here I've got a big T from dad and a little t from mom. Is that child going to be tall?
Yes.
Yes. Now I get a big T from dad and a little t from mom. Is that child going to be tall?
Yes.
Okay, well these kids are both going to get little t's. Are either of them going to be tall?
No.
So what are the odds in this scenario that they're having tall kids?
50%.
50%. And the other half of their kids are going to be? Not tall. Say that other word, not tall. Okay. I don't consider myself tall, but I also don't consider myself short. So, to me, tall. It's like 6'6". That's all. All right? What if it's recessive? Is dad tall?
Yes. No.
Is mom tall?
Yes.
Is anybody on this chart tall?
No.
If it's recessive, you need both. You got to have two capital T's. None of them have it. Could dad have found somebody else to have kids with and maybe have tall kids?
Yeah.
I know. It's like he's like, oh, that's so wrong. But what if I wanted a basketball team in my own house? Okay, let's stick with recessive, but let's say he met someone else, and it wasn't because of that genetic study they did. Things don't work out sometimes. Now he's met someone else. Are any of their kids going to be, remember, this is a recessive trait, are any of their kids going to be tall? What percent? 25%, because this is the only box I'm going to get two capital T's in. I'm going to have a big T, little t, big T, little t, two little t's. So 75% of their kids are still not going to be tall in this scenario, but 25% of them will be tall. Now, we don't do this for tallness, but we do this for things like Huntington's disease, sickle cell anemia, cystic fibrosis. There are, I think, 130 genetic diseases that they can pre-test mom and dad for, and then there are a bunch of others that they can follow up testing baby for before baby's even born. And they can even do this so early that if it is a severe disease, they can give the parents the option of a medical abortion. And that's an option. That's an option. Does anybody in here, actually I shouldn't ask you about your Huntington's disease, or your medical records. So let me just say it this way. Unless you have Huntington's disease, you don't know whether or not you would want a kid with Huntington's disease. So the only people equipped to decide whether or not they want to raise a child with Huntington's disease are parents who have Huntington's disease. And that's why those parents are given the option of, yes, this particular fetus has Huntington's disease, you have options. Now, is that okay in healthcare? Yes, I don't care. I don't care what you think is okay. In healthcare, is that okay? And if you cannot advise people thusly, you need to not work with people like that. Okay? A medical abortion is an option given to people. And here's another thing. You all know what anencephaly is? Anencephaly, anybody ever heard of that term? Let me tell you what it is. Baby with no brain. Baby with no brain. We can pick it up in the first trimester. If that baby's born, does that baby have a chance to live?
No.
No. It gurgles and grunts for a couple minutes and then it suffocates and dies. Okay? That's one of the first reasons we had medical abortions offered and covered by insurance for a mother that has a fetus like that. Because that mother should be able to terminate that pregnancy. Otherwise, she's going to give birth to a blob-looking baby that appears to be a monster and dies in her arms. That ain't fair. That ain't fair. So I guess I know where I live. And my point in kind of getting down this little road is in healthcare, there are things, have you all ever heard the term spontaneous abortion? What is that? What's another word for it? It's a miscarriage. It was going to happen anyway. It just didn't work out. Okay? And a medical abortion is an abortion for disease or defect, and sometimes just to save who? Mom. Because who's supposed to be the priority during pregnancy? Mom is the priority. Sorry. I know we love babies and they're cute, but if it comes to mom or baby, it is basically mom we're taking care of. You all are going to find this. Even though you may disagree, a teen's view, oh, well, the baby. Oh, of course, it's a baby. Okay? But you gotta some of you might need to hit a little bit of the reset button when you're dealing with these kinds of issues. Here's another one, ectopic pregnancy. What's that? Is that really a pregnancy? No, there's no gestation at work there. That's not going to be a baby. What is it? It's an injury. It's an injury to mom. And when somebody's injured, what do? We fix it. That's what we do with an ectopic pregnancy. We cut it out. That's not an abortion. Okay? So kind of be a little bit careful. If you have really, really strong feelings about a certain topic, you've got to be careful when you go into healthcare, because this is how it rolls in healthcare. And that's the way it should be, by the way. One last example, a 14-year-old girl comes in and asks for birth control. Do you send her off and call her a hussy, or do you give her birth control? Do her parents have to be there? Do her parents have to be there? No, they do not. You can give her 1 month of birth control and say, I suggest you discuss this with your mother or an older sister, or come back and talk to me about it. But what's that 14-year-old girl? First of all, we know she's probably having sex, maybe, okay? But what else is she acting like? And it's a responsible adult, yeah. Mm-hmm. Mm-hmm. Is the guy going to get birth control? No. So if you send her away without birth control, after she came to your clinic, embarrassed as she must have been, begging you to help her not get pregnant, and you say, you shouldn't be doing that, you little, I'm going to call your mother, okay? Then we don't want you in healthcare. You should not be dealing with patients. Okay? And again, you all are going to be trained by better people about this stuff than me. I'm just telling you and preparing you. You get out into the real world, you don't get to bring your moral beliefs there. Okay? You don't get to set the moral bar. You may have a moral bar, and you're allowed to set your moral bar in your personal life wherever you want it. But when you come into work at a healthcare facility, you follow the general accepted ethical and moral standards in that healthcare facility. And if you can't, you'll get written up, you'll get cited, and you might lose your job. As well you should. Okay? So now that we're all clear on that, let's talk about some diseases. What are we on? Where's my hand?
Dr. Myers?
Yes.
Does Kansas cover that kind of procedures, like so medical termination?
Absolutely.
Wait, because like one of my coworkers' daughter, I think had Turner syndrome for the baby, but they didn't cover it. They had to go to Colorado for it.
Okay, Turner syndrome is not necessarily on every state's list. Can someone with Turner syndrome live yeah, they can. The one thing with Turner syndrome is that they typically can't have kids. But they can actually live to 70. You actually surprised me a little bit, because I would have probably guessed Turner syndrome would be on most lists. But Turner syndrome is pretty benign compared to some of these others. And by the way, I gave you all a really extreme example with anencephaly. I mean, a brainless person, literally no brain in the vault. It's a cranium, but nothing in it. It's like a flat hot water bottle. That's a really extreme example. Again, there are like 130, actually that's the wrong number, but there are dozens and dozens of conditions that they will offer a medical termination for in every state in the union, and they should. Good, I would have to research a little bit about how frequent Turner syndrome is not. And another thing is you said covered by that insurance company,
No,
maybe?
They didn't cover it. They had to go to Colorado.
Okay. To get it done or to get it covered?
No, to get it terminated.
To get it, okay,
$20,000.
yeah. Well, you know what, that may be one of those borderline conditions, Turner syndrome. We're actually going to talk about that in a second. Okay, let's not worry about penetrance and expressivity. Let's do bar bodies. Bar bodies. I'm not sure how much you all remember from your female gamete studies, but what I can tell you is that in the girl reproductive cells, it's a little bit cutthroat. It's a little bit cutthroat. So here's the deal. With boy gametes, the 4 products of a meiosis division all have an equal chance at fertilizing an egg. They all have an equal chance. In girl gametes, there really aren't 4 products of meiosis. After the first meiosis division, you have 2 potential eggs. And then a supervisor comes into the room and chooses the pretty one. You pretty, you not. And the not pretty one dies. And the pretty one gets to divide and do the second meiosis division. So we have 2 more potential girl cells again. Back in comes the supervisor and chooses the pretty one. It's not, I know, I'm being a little bit politically incorrect with this. But literally, one is chosen. The healthiest, juiciest one is chosen, the other one dies. And this is taken to an extreme for all of you women who have 2 X chromosomes. Each cell that is born with 2 X chromosomes, very quickly a decision is made that one of those X chromosomes will be used and the other one will not. It isn't necessarily the X chromosome that you got from your mom is always used and the one that you got from your dad is not. Or the one that you got from your dad is used and your mom is not. It's just kind of random, but in each cell, it's kind of like this cell ain't big enough for 2 X chromosomes kind of deal. And one of the X chromosomes is just told, we don't need you. And it just kind of fades away to the edge of the nucleus. I can't believe they did that. And it shrivels up. And it's just this little dot that you can sometimes see with a microscope. That dot is a bar body. And what bar bodies are inactive X chromosomes in female cells. Again, town ain't big enough for 2 of them, kind of thing. Finally, a mutation is simply a change in the base sequence on DNA. That's just a fancy way of saying you screwed up the recipe for that protein. Okay, that's what a mutation is. Let's look at 4 conditions. These are really our first real pathologic conditions. We're in our 3rd night together, and we're finally doing some pathophys, right? Our first disease is Down syndrome. Someone mentioned this earlier. It's also known as trisomy 21. Trisomy means 3 copies. So this disease is basically 3 copies of chromosome 21. How many copies of chromosome 21 are our cells supposed to have? 2. And in trisomy 21, everything else is right. They got 2 chromosome 1s, 2 chromosome 2s, etc., etc., etc. But when we get to chromosome 21, there are 3 of them in that cell. That causes an off-balance. This is far and away the most common aneuploid disorder. And if you remember, aneuploid means off by a factor other than 23. We're just off by 1 here. I want you to remember, and let me be careful here. And I don't know. I may go back and change this in my handouts. In a healthcare context, when we are speaking appropriately about healthcare, we are still allowed to use the term mental retardation. Okay? You'll hear it commonly used in talks and lectures. We are trying to back off it. Well, you all probably know cognitive impairment is typically what we want to replace with. That will eventually become an ugly term too, because that's what people do. They make these terms ugly so that in about 30 years we've got to change it all. Okay, so that's on people, totally. But I've got the abbreviation MR in my handout in this section, which stands for mental retardation. And for those of you who have studied this, or cognitive impairment, as most people will call it now, there are levels of this, degrees of this, if you will. I don't want to get into distinguishing those. What I want you to do is pay attention to which of these conditions tend to be associated with mental retardation and or cognitive impairment, and trisomy 21 is one of them. Also, definitely remember these kids are at high risk for congenital heart disease and leukemia, to the point that they are typically checked annually for things like murmurs, coronary artery disease, and they get a CBC every 6 months, I think, just to make sure their blood counts are good. Heart disease and leukemia. How many total chromosomes? 47 per cell. That's one too many. One too many. Trisomy X. You thought 2 X chromosomes was too much. Try 3. That's one crowded room. Women with 1 extra X chromosome. Everything else is fine. They're autosomes. They got 1 from mom, 1 from dad. Everything lines up great. But 3 X chromosomes. Many of these women look normal, but they tend to have slight cognitive impairment or mental retardation, and they tend to be sterile. Their gametes just don't progress well enough. Not sure they know why yet. Because when they study these patients, they find that many of them have 2 bar bodies. So the shutting down of all but 1 X chromosome works in most of their cells. That's why they're phenotypically normal. But for some reason, something goes all wrong with the gametes in their division. So that's why they're typically sterile, and they typically have cognitive issues. Turner syndrome was brought up here. These are women. So by the way, how many total chromosomes in trisomy X?
47.
Per cell. Good, 47. Okay, Turner syndrome. This is the opposite. This is women who have only 1 X chromosome. And you would think, after what I said about bar bodies, you would think, well, that should be fine, because 1 is going to be cast off anyway. Sorry, it doesn't work that way. You need to have 2 to start for everything to work right. Turner syndrome patients, this is the one I would want you to mark as being less associated with mental retardation. In fact, many patients with Turner syndrome, they're cognitively perfectly normal. They become accountants. Okay? That's a math field, so you all get the point. Okay? They, however, do tend to be sterile. They can't have kids. They tend to be short. And they tend to have this webbing of the neck. Like their neck is a little wider and it kind of, the skin kind of flares out, kind of angles out down towards their shoulders. In this case, you don't have to worry about it. You can Google images of, you all are the Google generation, so anything we talk about, Googling images, you actually visually is a great way to learn some of this stuff. I will warn you, there are a few conditions we talk about that you kind of want to be careful what you Google. I would even put that anencephaly, the one that I mentioned. If you don't have a stomach for it, don't Google it. Don't Google images of it, especially if you love kids. It can actually be a little bit traumatic. I say this because about 8 years ago, when I first did this class, I told a whole class of people to go Google stuff like anencephaly and cyclopsia. Do you all know what cyclopsia is? Fusion went wrong, so there's one big eye in the middle of the head. Those babies don't survive. I said, oh, go Google it if you don't believe me. And a couple of women came back the next night, they were really upset. You should have warned us about that. I was upset the whole night, I couldn't sleep. Those poor babies. And you know what, I get it. It's not fun to look at. It's not fun to see. But healthcare, you kind of got to toughen up a little bit. Maybe not in that way, but you got to toughen up a little bit. So Turner syndrome, how many total chromosomes in their typical cells? 45. Good. This might be what I asked. Like I might say, which of the following has 45 chromosomes in most cells? And I list Down syndrome and trisomy X and Turner syndrome. You want to jump on that third one, okay? Klinefelter syndrome is boys with 2 X chromosomes. They have a Y, but they have 2 X's. And this usually leads not only to some mental retardation. Do you all know what gynecomastia is? Anybody ever heard of that? Yeah, we can say it. Boy boobs. Boy boobs. Basically what this is feminization of a male. Because of the extra X chromosome, there are kind of some female features. Like gynecomastia, they tend to be sterile, sparse body hair, and poorly developed boy parts. Okay? 47 total chromosomes in this condition. So there are 4 conditions that are all aneuploid conditions. There is a wrong number of chromosomes, but it's only off by 1. It's not off by a factor of 23. This 5th one, I'm not really good with French, but I think it's
Creedy shot.
Thank you. It's that. Creedy shot. Here's it. Yes, that's it. Yep, that's it. The foreign languages I learned were Russian and German, so that makes it even worse trying to speak French. They're very hard compared to French. Cri du chat, or cry of the cat. This is not a wrong number of chromosomes. These patients have 46 chromosomes in their typical cells, but there is a partial deletion of chromosome 5. So it's just a piece of it is missing. This is a very severe disease, severe mental retardation, microcephaly, that means very small head and brain, heart defects, and these kids tend to cry like a cat. Very good, that's why they call it that. I'm not going to imitate it. Again, it's probably something you could Google. 5 diseases, questions on those? The stuff that I emphasize or mentioned is if I skipped it, I won't put it on the exam. Okay? All right then. Yeah?
What's the lifespan for the
Cri du chat? Low. It's low. With management, they just need long-term care, permanent care. I have a cousin who was suffered an anoxic brain injury at birth, and I think she's 2 years older than me. Okay, she has never been able to talk, never been able to feed herself. There was a point at which she could walk enough to kind of push a wheelchair, so the wheelchair is kind of helping hold her up. So this is severe mental retardation, is what the diagnosis my aunt was given when she was born. She's been in a home her whole life. She's had to be fed and cared for and looked after. My aunt, who is now going on 80, still goes to see her twice a week and won't leave the hometown because she's not leaving her daughter. Makes me kind of sad, because I'm like, when I've even asked my mom, when Aunt Janice dies, who's going to visit Gwen? She may still be there. But that's how these kids tend to end up. It's severe enough that they need long-term, all-day, inpatient care, special needs care. Yep. These other diseases, not so much. In fact, a lot of these other diseases, do you all know there's people with Down syndrome who have jobs? Families? Yeah, they go to school. Okay. Yeah, there's levels of severity. Do you know that Down syndrome is one of the diseases you get to elect for a medical abortion if you don't want to raise a child with Down syndrome? And do you know that when given the option, less than half of parents abort? It's like, I think 42% of them choose to terminate, 58% of them choose to have the kid, and a very, very small, tiny, negligible fraction of those parents have Down syndrome. So you're talking pretty much about parents who do not have Down syndrome are confronted with their child, their unborn child, does have Down syndrome. 58% of them say, I still want to raise that child. That's our kid, and we're going to see to that child's life. And 42% say, I don't think I'm equipped to deal with a Down syndrome child, especially if it's severe. So we'll terminate and we'll try again. Okay? Neither side is wrong. Neither side is wrong. Okay? Yeah.
What is Huntington's disease?
Getting there. Getting there. I think we'll get there tonight. We're going to be pushing it, but I promise we'll get there. Okay? And the quick answer is it's a genetic neurologic disease. Okay, so now I've at least left him with an answer, and we'll get there. Okay, this section here, the reason I'm not sure we'll get into the ins and outs of Huntington disease tonight is I really want to take the time to make sure you all get these 3 sections at the end of page 3, because these are tough. This is probably where a lot of you are going to spend a good amount of your time. They are the 3 types of inheritance, disease inheritance. And this is not for Down syndrome and stuff. These are actual mutations on one part of a chromosome that can be passed down to your offspring. Okay? So the kids that have that are born with these diseases, all of their cells have the right number of chromosomes. They've all got 46 chromosomes and 23 in their gametes. Okay, everything is cool. It's just that one of their protein recipes is off. So the first pattern of inheritance is called autosomal dominant, or AD, autosomal dominant. Autosomes are chromosomes 1 through 22. Dominant means you only need one allele to have the trait, or in this case, disease. Autosomal dominant. So that means it has to be on chromosome 1 through 22. This can't affect my X or Y chromosomes. And all I need is one from mom or dad, and I'll get the disease. Okay, now what this means is there are no skipped generations. Here's what no skipped generations means. It means that if you have an autosomal dominant disease and you look up your pedigree at your parents' generation and your grandparents' generation and their grandparents, you look up at every level somebody had this disease. So this disease does not skip generations. Okay, that's what that means. Affected heterozygotes pass the trait on to half of their children. Affected heterozygotes. Let's say the gene for this autosomal dominant disease is D, big D, and the healthy gene is little d. If these are the two choices I've got, does it make sense that I've got a 50-50 shot of passing the disease on to my offspring and the same shot at passing the non-disease? Okay, and if I pass this one on to my offspring, does it matter what the female in this? No, it's a dominant disease. That child is going to have it. Huntington disease is our example. It is the most talked about and discussed autosomal dominant disease. It has to do with a mutation on chromosome 4. You should definitely remember both autosomal dominant and chromosome 4. And it is a slowly deteriorating neurologic disease. It's kind of like an accelerated multiple sclerosis, which often leads to death in the 20s. Some people live a little longer, and I'm sitting here thinking we're going to talk more about it, but Christian, for the more details, we got to wait till Unit 3 when we're in the nervous system. Hopefully we've at least sated you for now. Cool on autosomal dominant. Okay, autosomal recessive. Again, we're talking about chromosomes 1 through 22. And in this case, to have one of these diseases, you need both of your parents to give you the disease gene. So in this case, if we're talking about autosomal recessive disease here, this person is a carrier. They don't have the disease because they didn't get the second hit here. They've only got one disease gene. But they can pass that disease gene on. And that's, quote, what the carrier state is. For autosomal recessive inheritance, these are also fairly rare when you look at the whole population. Just like the previous, men and women affected equal. Skipped generations are common. That again means I might have an autosomal recessive condition, but it turns out my parents and my aunts and uncles, none of them have it. But great-grandpa had it. Okay, so that's what I mean by skipped generations. Consanguinity. Do you all know what that means? Anybody know what consanguinity is? Say that again? Well, yeah, you're not going to like it. It's actually inbreeding. And inbreeding, I know we all think of the movie Deliverance. Is that it? Oh no, there's one for your generation too? Well, it was Deliverance for my generation. And kind of a bad name for West Virginia. It turns out it was filmed in Georgia. So anyway, consanguinity is not as bad as it sounds. There are a lot of very, very, very rural communities across the world, including in the United States, where it would not be that out of the ordinary for someone to get married and have kids with the love of their life and later on find out, oh my God, that's your third cousin. And I know, we all are like, that's like a horror movie. But it actually isn't quite as twisted as you think. We're talking about rural populations where there aren't that many different families contributing to the gene pool. And we tend to find autosomal recessive diseases in such situations. That's what consanguinity means. Affected persons, so that means a person with an autosomal recessive disease, may have affected brothers and sisters, but rarely has parents with the disease. And the reason for this is these autosomal recessive diseases tend to be bad enough that you don't end up having kids when you have the disease. So the parents that are carriers, they don't have the disease. They might pass it on. But autosomal recessive diseases tend to leave people not having kids. Question?
Since inbreeding the same option of that, is it caused because of the inbreeding?
It isn't. It just makes, the only reason the quote consanguinity contributes to that is because there's simply a higher odds that if I'm a carrier, then somebody in my family is also a carrier. So that if I end up, even if it's with a third or fourth cousin, having kids with a third or fourth cousin, it's a little bit more likely that we will have both be carriers than if I randomly had kids with someone. Definitely a fair question. It doesn't cause it, it's just really kind of a statistical consequence. Offspring of carrier parents. So here's a carrier parent in an autosomal recessive disease. Offspring of this parent have of carrier parents. So both my parents look like this. They have a 25% chance of having kids that have the disease. We just did that in a Punnett square for Thomas. Okay, if this is mom and this is dad, okay, now if I did my little Punnett square right here, this would be 25% of my kids. Does that child have the disease? Yeah, are any other of the kids getting both big Ds? No. So while I might have some carriers, only 25% of our kids, again, if both parents are carriers, 25% of their kids are going to have the disease. And an example disease here, again, this is another one we'll talk about later on when we get to the lungs and the digestive tract. This is no fun to see in its worst form, cystic fibrosis. There are absolutely worse diseases out there. I saw this personally during my training, this among a couple of others, and just trying to suck this thick mucus out of these poor kids' airways kind of traumatized me a little bit, because I just thought this poor 4-year-old did nothing to bring this on, and they're suffocating in their own lung mucus. That just sucks to see. And you know what? So does childhood leukemia, and so does muscular dystrophy. So there's a lot of stuff that really, really sucks to see, but I think even somebody like me is a little bit more sensitive when it's a child. So remember, cystic fibrosis is our example of an autosomal recessive disease, and it's a mutation of chromosome 7. Yeah?
Sorry, you guys have questions, but can you tell in the lupus the child with cystic fibrosis?
Yes, cystic fibrosis, Huntington's disease, most of the major, they do now 2 procedures. They may have added to this. Remember, I've been a teacher for 10 years, so and I don't read up on everything, but they can do both an amniocentesis and what's called chorionic villus sampling. And basically what that is sampling is the fluid around baby and the blood in the baby's umbilical circulation. And with those studies, they've gotten much better at being able to definitively say whether a child has this condition or not.
Is there any sign to make you do that though?
Being a carrier, being a known carrier, and most people learn they're carriers when they're told, oh, your great grandma had cystic fibrosis. And then you're like, well, none of my parents or uncles had it, so that's why they never really talked about it. Now, I could be a carrier, and I don't know if the person I'm having kids with, and then they might go and get some genetic counseling, or even talk to an obstetrician and say, oh my gosh, we're pregnant, but we don't even know if we're carriers of it. So that's usually how it kicks off. Are we going to catch everybody by screening? No, a lot of people are going to have kids, and this is going to happen, and that's why we want to get better and better at obviously treating it. They're doing lung transplants now for cystic fibrosis, and apparently it's working in a lot of kids. It used to be, if you would have asked me 5 years ago, what is the most common condition they do a lung transplant for, I would have told you pulmonary fibrosis. They don't do lung transplants for lung cancer. Lung cancer is pretty much not treatable. Okay, they try to radiate you, and then they say you're going to probably die soon, so you may as well just keep smoking. Really, that's how a lot of doctors approach it. But lung transplants for pulmonary fibrosis actually worked really well, where now I think they are doing more lung transplants for cystic fibrosis now than any other condition because of the fact that it's working well. They're having some very, very positive results. Again, I don't know all the ins and outs, how old you have to be. It's really, really complicated surgery, but that's encouraging to know. Okay, autosomal recessive. Let's do X-linked inheritance. This is diseases passed on the X chromosome. There's really no Y-linked inheritance to speak of because, sorry guys, the Y chromosome, it really doesn't do diddly. All it does, very early in our development, when we're about, 512 cells big, it turns on the boy button. Boy. And that's pretty much it. That's pretty much what the Y chromosome does. Okay, we're going to be a boy. It makes that determination. We have a chart downstairs in our A&P room that lists the known functions of every chromosome. And I'm pretty sure that the busiest chromosome is X. It's either chromosome 1 or chromosome X. And I'm kind of afraid to count it up and see because I feel so insecure looking at how little the Y chromosome does on that poster. X-linked inheritance. This is way, 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 way more common in men because we only have one chromosome. And if it is mutated, we don't have another choice. In girls that get a mutant X chromosome, that usually becomes the Bar body. The good X chromosome takes over in that cell. It is never transmitted father to son. Why? Yes, dads don't give their sons an X chromosome. That's why every situation, fathers give their sons a Y chromosome by definition. That's the only place a Y can come from. Skipped generations are common here. These actually act like recessive diseases. And affected fathers. So think of a male who has one X and one Y. They have the disease. Affected fathers. They will pass that gene on to all of their daughters because every daughter I might have is going to get my X chromosome. I've only got one to give them. And then those daughters are going to pass that gene on to half of their sons. Let's see why. Here's me, or let's not make it me, but here's the person with the X, a male with an X-linked disease. That's why I put the asterisk on it. If this person has a daughter, the daughter has to get this chromosome. But she also got probably a good X from her mom. And now, when she decides to have kids, and her kids are boys, there's a 50-50 chance she'll pass this disease on to the boy, and a 50-50 chance she won't. That's what that sentence says. Everybody clear?
So if your body makes one of the bad X's of the bar bodies, can you still pass it on?
Bar bodies cannot be passed on, and even if the material that makes up a bar body is passed on, it is just debris. Okay, so it has no function. And I don't want to be like 100% about this, but in general, a diseased X chromosome in a female cell will become the Bar body. It's almost like the supervisor knows something's wrong with you, so you go for that. I know that's, again, it sounds so mean. You're the pretty one. That was really a bad way to describe it. I apologize. But it gets the point across, right? So, but I think that's a really good question. And in most cases, a defective X chromosome in an otherwise normal female cell, that defective X chromosome will become the Bar body. And that's why these kinds of diseases are really rare in women. And your example here, another bad one, Duchenne muscular dystrophy, also known as DMD. You all probably don't remember, they used to have these Labor Day telethons when all the TV was canceled for this muscular dystrophy telethon, trying to raise money for kids that have muscular dystrophy. These kids, it's basically ALS, in Lou Gehrig's disease in childhood. Their muscles around their hips and their legs just stop working. In order to stand up, these kids literally will use their arms to push themselves up. By the time they're 12 to 15, they're in a wheelchair. By the time they're 20 to 24, they're dead. So Duchenne muscular dystrophy, why didn't I put a chromosome there for it? Because it's the X chromosome. That's the chromosome. It's under X inheritance, so that's the chromosome. Remember, the other two sections were autosomal. It could have been chromosome 1 through 22. Spend a little bit of time reading through those sections. We actually kind of gun through the rest of this handout. It's a little bumpy at the top of the next page, but we're going to really fly through the next part of this handout. Read through those. Make sure you can kind of reiterate some of the important points I told you about autosomal recessive diseases. Also, I have two of you, I think, that came in after I called roll. Please come up here and at least show me your name or add it to the list. And Denver, just a couple comments for you. Anybody have other questions? Please be careful going home, and we will see you all again whenever our next night is, Thursday night. Man, I'm having a hard time about this year. Don't forget your phones.
I want to visit with you when you're done.
Hi.
Okay, so on this, you just want us to know the different membrane transports or all of it?
Thank you.
You know,